the 11th environmental lecture presented by the environmental group of the American Chemical Society Pittsburgh local section. I'm your host, Ron Hong Lin, member of the ACS Pittsburgh local section and the chair of the environmental group. Okay, the environmental group is a topic group within the local section, and we support various activities, including this monthly environmental lecture series to promote a deeper understanding of science, technology, and our environment. The lecture is being recorded and will be available online later. I'd like to remind everyone that the views and opinions expressed in this and any future lectures are solely those of the presenters and do not necessarily represent the views of the ACS Pittsburgh local section. And our speaker today is Dr. Yujie Men, Associate Professor in the Department of Chemical and Environmental Engineering at UC Riverside. Dr. Men earned her BS and MS degrees in environmental engineering at Tsinghua University in China and a PhD degree in civil and environmental engineering at UC Berkeley. And she did her postdoc research at UC Berkeley and EWAG in Switzerland. Before joining UCR in 2019, she was an assistant professor at the University of Illinois and Urbana-Champaign. Dr. Men was a recipient of the prestigious NSF Career Award in 2021. She currently serves as the editor of Water Research X and the Journal of Environmental Chemical Engineering. Dr. Men's research focuses on the interactions between microbes and the environment impacted by the contaminants of emergent concern. So today, she's going to talk about her recent work on structural specificity of microbes transformation of PFAS and its implications. With that, please join me and welcome Dr. Yujie Men. And thank you everyone for joining us today. And I hope you enjoy the lecture. Dr. Men, you can take over and share your screen now.
Thank you very much, Dr. Lin, for the invitation and the nice introduction. Let me share my slides now. Are you seeing the slide okay?
Yes.
Great, thank you. Okay, today I'm going to talk about the structural specificity of microbial transformation of PFAS and its implications. First of all, I would like to give my great acknowledgement to all my lab mates, lab members, my great collaborators at UC Riverside, University of Minnesota, and the industrial partners, as well as the funding agencies. Without them, none of my work could have ever been done. So I would like to give the acknowledgements to them at the beginning. Okay, PFAS. I think nobody is not familiar with this PFAS term, which represents per and polyfluoroalkyl substances. And in short, that's PFAS. They're considered as forever chemicals. Structure-wise, they have a structure of fully fluorinated carbon, CF3 or CF2, in connection with a non-halogen or non-hydrogen bond. So there are many phases of PFAS compounds. The most commonly ones are the PFAAs, perfluoroalkyl acids, and some of them, actually 6 of them, are regulated by EPA just this April. And EPA has established the maximum contaminant levels for those 6 PFAAs. And there's another whole group of PFAS compounds that can be transformed in the environment to PFAAs. We call them PFAA precursors. They have been widely used in different applications. In our everyday life, we have this oil repellent, water repellent materials, textiles, and also those plastics. And also in industry, there are firefighting foams. There are engineered oils or engineered liquids. Those are all containing PFAS compounds, different types. So literally they are everywhere. And then they are considered as forever chemicals because their physical chemical structures make them very resistant to environmental conditions. They're really recalcitrant to environmental degradation. That's why we consider them as forever chemicals. And let's take a detailed look at these organofluorines. Actually, all of the PFAS compounds that I'm talking about right now in the previous slide, they are all man-made. They are all man-made, synthesized chemicals. And in this slide, I want to give a comparison between the nature-made organofluorines, those are naturally occurring in nature, versus the PFAS molecules that are all man-made. And they were invented back in 1940s or so. So they haven't been introduced to nature very long. It's less than a century. So on the left-hand side, it's a nature-made organofluorines. And I'm giving you one common example, which is fluoroacetate. This molecule has a monofluorine substitution in an acetate structure. It is produced by plants. And why plants produce this chemical is they use it to fight against their bacterial pathogens. Those pathogens may infect plants. So they're using this fluoroacetate to fight against the bacteria. This fluoroacetate is toxic to bacteria. And during millions of years of evolution, bacteria evolved this mechanism using this enzyme called 2-haloacid dehalogenases, HAD, to degrade fluoroacetate by removing the fluorine and replacing with a hydroxyl group, forming glycolate, which is non-toxic to the bacteria. So in this mechanism, bacteria can detoxify fluoroacetate. So that's what's occurring in nature. And on the right-hand side are representative PFAS structures, man-made PFAS structures. You can tell the major difference is there are many, many carbon-fluorine bonds there. And for many compounds, especially PFAAs, all the carbon is connected to a carbon-fluorine bond. So all the carbon-hydrogen bonds are replaced by carbon-fluorine bonds. And why they are so strong, they are so recalcitrant to degradation, we look at the carbon-fluorine bond property, we can tell the carbon-fluorine bond is the known strongest single bond in nature, and it has the highest electronegativity. In other words, when we want to break it down, we need a much lower redox potential. And that redox potential may be within the limitation of the physiological limit of any organisms due to the low redox potential. And the last property is that even if an organism would be able to do the deformation in their cells to do the detoxification and chop off the fluorine from the carbon, the formed fluoride ion is toxic to cells. That's well known. And then that cells must have a detoxification mechanism to also expel the fluoride, the toxic fluoride from their cell too. So those are the facts that prevent PFAS compounds being degraded easily in nature. So the question is, can we find certain microorganisms in nature that have powerful defluorinases to break down the carbon-fluorine bonds? And if we let the nature develop such enzymes, it may take a long time. Given its short time period after the invention, and the microbes in nature are not exposed to PFAS compounds for a long time. They haven't gotten enough time to develop this PFAS degradation mechanism or enzymes. And some of you may notice that the PFAS molecules may be effectively degraded by certain physical-chemical approaches. But the question is why we still care about the PFAS biodegradation. I have two reasons from science perspective and from an engineering point of view. Scientifically, the carbon-fluorine bond cleavage by organisms, especially by microorganisms, is very understudied. It's still very unclear about the biotransformation pathways and the responsible enzymes. And the molecular mechanisms is still unknown. And from an engineering perspective, we want to understand better. After PFAS is discharged in nature, in ecosystem, how they get transformed in environmental condition, their environmental fate. And also, if we could discover any effective microorganisms that can degrade PFAS, we can develop and apply in situ remediation technologies, which would be still ideal to treat PFAS on site, in situ, without pump and treat. And again, bioremediation technologies are considered more cost-effective compared to other physical-chemical approaches. They also environmentally friendly. And once we understand the mechanism of microbial defluorination, we may also design certain more biodegradable alternative PFAS. Because in some areas, like medical devices, electric devices, and aerospaces, these application areas, some PFAS molecules, they are still not replaceable. That means they're still not suitable surrogates or substitutes of PFAS. We still need PFAS, but we want PFAS to be less persistent and more biodegradable. And those are the reasons why we still care about PFAS biodegradation, and we want to answer these key research questions. First, since there are thousands of PFAS structures that's been manufactured, which of those PFAS chemicals, PFAS compounds, could be more vulnerable to biodegradation? Via what pathways? By which microorganisms? And through what molecular mechanisms? So what I'm going to talk about is the cleavage of the carbon-fluorine bond in the PFAS structure. So what we did is we selected environmental microorganisms. We chose two conditions, anaerobic and aerobic. Anaerobic conditions, we chose a dechlorinating microcosm, which is commercialized, that's KB1, and has been widely applied to remediate chlorinated solvent contaminated sites. And another type of representative anaerobic microbial community is the anaerobic sludge, because it is commonly occurring in wastewater treatment plants. The third one we used was the groundwater from AFFF impacted sites. AFFF represents aqueous film form foam, aqueous film form foam, and that contains PFAS. PFAS is one of their formula. And in aerobic conditions, we selected a representative microbial community that is the activated sludge. And we use those different microbial communities and we spike in our target PFAS molecules and to compare the biotransformation activities and trying to understand the structure specificity of PFAS biotransformation. The first PFAS structures we tested are perfluoroalkyl and saturated. Those are the commonly occurring and the most concerning ones, and those are 6 of them are regulated. These 2 is among the 6 compounds regulated by EPA, PFOA, and GENEX. Unfortunately, using our investigated microbial communities, the 4 different types of communities, we didn't see any transformation by those microbial communities under either anaerobic or aerobic conditions. And the next is those are the structures that's not working in our tested systems. And for those that are working, the first one is We further tested the per and polyfluoroalkyl unsaturated structures. So the difference is that there is carbon-carbon double bond. And these type of structures underwent reductive defluorination by specific microbes in anaerobic communities. In a recent paper we published in Science Advances, we identified the specific anaerobic microbe, Acetobacterium species, that were capable of reductive defluorination of unsaturated PFAS structure. We used this representative structure, PFMPA. It has 6 carbon. It has a carbon-carbon double bond next to the carboxyl group, and it has a branch there. So the carbon-carbon double bond plays the most important role to make this structure more vulnerable for microorganisms. And the pathway we confirmed is via 2 pathways. The major pathway is a reductive defluorination pathway, replacing this fluorine with the hydrogen. And then the other pathway is the other pathway is highlighted in blue is the hydrogenation pathway, adding 2 hydrogens to the double bond, forming hydrogenated product. So these are the confirmed transformation pathways using high resolution mass spec and also 19F NMR. We further tested other unsaturated PFAS structures, including the 622FT-UCA, which is a transformation intermediate of 622FTCA, and all of them showed biotransformation activity and defluorination too. We further studied, why this could occur and what are the molecular bases for the reductive defluorination. We tested 5 different Acetobacterium species and to compare their defluorination activity. And we found there's one outlier here, that is the Acetobacterium fermentarium. It doesn't show any defluorination activity at all. And when we compare their genome, we found this species, this fermentarium species, doesn't have an intact fluorine transporter gene. Its second component in that operon is trunked. That makes it unable to expel the fluorine ion out of the cell. So once the fluorine is formed in a little bit amount inside of the cell, it will toxify the cells and seize every activity. So we attributed the no defluorination activity of this fermentarium to the lack of active fluorine exporter. So an active fluorine exporter is very essential for intracellular defluorination. Second, we went one step further to look for potential enzymes that are catalyzing the reductive defluorination. And our study implicated in the electron bifurcating cathode reduction pathway being involved in the reductive defluorination, because we looked at the structures of PFMEUPA here with a double bond. We looked into the genome of the Acetobacterium species, and we found this cathode also has a double bond, and it undergoes reaction via the electron bifurcating enzyme complex. So we hypothesized that this cathode reduction pathway is involved in the PFMPA defluorination. And how we tested it is the first we use cathode inhibition study. We add cathode to the cell culture, and we compare the defluorination activity between the cell cultures with cathode addition and without. The solid lines indicate the cell cultures with cathode addition. And you can see the pairing compound decay ceased during the cathode was still present. And once the cathode is reduced completely and the reductive defluorination starts to occur, the green curve represents the fluorine ion formation. And the dashed curves is our positive control, which doesn't have caffeine in it. This is the caffeine inhibition experiments indicate caffeine may inhibit the PFMPA deformation by competitively binding to the active site of that deformation enzyme. And our collaborator, Professor Lawrence Wackett from University of Minnesota, helped us did the enzyme modeling and finding that the PFMP CoA form bind nicely into the caffeine reductase binding pocket. So that indicates these two structures is actually very similar with each other. So they may bind to the same active site of the caffeine reductase. And then the caffeine reductase is highly likely involved in the reductive deformation. And we're still collecting more direct evidence via biochemical or in vitro assays. So these studies are still ongoing. That's about unsaturated PFAS. So the next group of PFAS structures we tested is the chlorinated PFAS. And we found that the introduction of carbon-chlorine bonds is helpful to make the molecule more biodegradable. And how they get biodegraded is they are degraded via dechlorination facilitated defluorination in anaerobic conditions. And we do also isolated anaerobic species that are capable of this reaction process. And the chlorine PFAS is not uncommon. Actually, they represent quite a large group of compounds. One example is the PCTFE. It is a sister of Teflon, PTFE, and its derivative CTFE oligomers. They are used as engineered fluids in different applications too. And we took CTFE oligomer acids, 3 oligomer acids with different chain length, and with dimer acid, trimer acid, and tetramer acid. We also tested some PFCA precursors and their alternatives, such as 5-carbon, 7-carbon, and 9-carbon terminal chlorinated PFCAs. What we found is that in anaerobic conditions, the CTFE dimer acid, trimer acid, tetramer acid can all be fastly degraded by anaerobic microbial communities. As the chain length goes longer, it is more biodegradable, and the total deformation can reach up to about 80% within like about a month or a month and a half. So why it caused this molecule so biodegradable is we attributed that to this anaerobic microbial dechlorination process, which is the first step that triggered the downstream deformation reactions, especially for the third type of microbial dechlorination, which is hydrolytic dechlorination, replacing the chloride with the hydroxyl group, forming unstable fluoroalkyl structures. And this fluoroalkyl structures is really, really unstable and can undergo spontaneous defluorination, forming fluoride ion and diacids, or by carbon-carbon bond top-off cleavage and forming chain shortening products, and which continue to be defluorinated until the end. So through this pathway, from the very right pathway, this original molecule could be potentially completely degraded. And then we isolated anaerobic microbes and identified 2 of them, Spermosa and Desulfo vibrio species, that were capable of the CTFE oligomer acids dechlorination and defluorination. They perform similarly, and also with slight difference is the Spermosa tends to form unsaturated structures products, which is not that favorable to the downstream deformation. As you can see, this eliminative dechlorination forming unsaturated products, but unsaturated products undergoes very minor deformation downstream, whereas the disulfuribral species prefers this hydrolytic dechlorination forming intermediates that are more favorable to the abiotic deformation pathway. So that's why the total deformation, the very left column, orange color, disulfuribral exhibited 77% of the total deformation, with spermosa has showed a lower total deformation percentage. So their pathway is different, indicating the responsible enzymes and the molecular mechanisms are different between the two isolates. Both are capable of CTFE oligomer acids defluorination, dechlorination. So the identification of enzymes in the different microbial species are still ongoing. The fourth types of PFAS structures we tested are polyfluoroalkyl, saturated or unsaturated, doesn't matter, polyfluoro structures. Polyfluoro structures, the meaning is that on certain carbon atom, there is still carbon-hydrogen bonds. So the key is the carbon-hydrogen bonds is more vulnerable to oxidation reactions, and it's a weaker attacking point for aerobic microorganisms. So we found that aerobic activity sludge communities in aerobic conditions, they can do the polyfluoroalkyl structure degradation and transformation. And we used structurally similar compounds, polyfluoroalkyl carboxylate acids, with different chain length, carbon 3, carbon 4, and carbon 5. And they all have similar structures with the terminal CF3 moiety, just different by the chain length. Interestingly, we found the odd number chain length showed very high biodegradability and deformation percentage, whereas the one in the middle with 4 carbon, there's very little transformation activity and there's no deformation occurring. And we are trying to explain why, and we identify the transformation products, and we hypothesize this beta oxidation-like pathway, because this can very well explain why the odd chain number of this structure showed great degradation, whereas the even number didn't. So via the transformation product analysis, we found we proposed the transformation pathway by the beta oxidation-like, and there's a HF elimination at the beginning, similar as the beta oxidation of the fatty acids, and then with an oxidation at the beta position and chopping off 2 carbon at a time. 2 carbon at a time. So that can explain why the C4 structure didn't show any degradation, is because if the cleavage happened 2 carbon at a time, by leaving 1 acetate, the remaining will be trifluoroacetate. So the trifluoroacetate is very known and resistant to any biodegradation. And that explains why the C4 structure didn't show any degradation, whereas the C3, this is the complete degradation pathway we proposed. And the C5 is when you chop off 2 carbon, leaving an acetate, remaining the remain part is the same as the C3 structure that could undergo a complete degradation pathway as we proposed at the bottom. All right, and then the last group of PFAS we investigated and observed biodegradation is the ether PFAS. So ether PFAS is also commonly used. One example is GenX. GenX is the substitute of PFOA in some applications. And then there are also other structures that's similar to the GenX structure. We want to know whether the introduction of the ether group could enhance the biodegradability of that molecule. So what we found is that ether PFAS can undergo oxidative and hydrolytic O dealkylation by aerobic communities such as activated sludge. The thing is, there's only one requirement is the ether PFAS has to be polyfluorinated because we didn't observe any biotransformation activity for perfluoro ether PFAS. As you can see from this figure, this one, if the ether group, once both sides of the ether group is fluorinated, both sides, the carbon on the ether, both sides of the ether group is fluorinated, there is no deformation such as E2, E3, and E8. As long as there's one side of the ether bond has non-fluorinated carbon, this part is more vulnerable to biodegradation and will be attacked first. And the biodegradation activity on this non-fluorinated ether side will trigger downstream abiotic deformation. So that's our interpretation. So the oxidation will occur to the non-fluorinated carbon, such as this alpha carbon for this structure, and via oxidation and in OH. And then this is a hemiacetal structure, which is unstable and will undergo spontaneous hydrolysis in aqueous phase, forming this alcohol, fluoroalcohol structure. Again, this intermediate is transient, it's unstable, undergoing spontaneous defluorination, and also forming non-fluorinated oxalate as a product, which goes to the central metabolic pathway. And then the oxidation of this E5 structure similarly occurring to the carbon next to the ether group by adding OH, forming hemiacetal, and undergoing hydrolysis and spontaneous deformation. And then we further investigated the combination of those moieties we identified, such as unsaturated carbon-carbon double bond and ether group. So the two together, see here the structure E12. E12 showed great biotransformation activity too. And although the ether group, both sides have fluorinated carbons, but one side also have this carbon-carbon double bond. So the presence of carbon-carbon double bond increased the biodegradability, even though the other carbon next to the ether is also fluorinated, fully fluorinated. So with the two together, this structure underwent hydrogenation, which is a major biotransformation pathway by adding 2H to the unsaturated double bond. Besides this major non-defernating pathway, there are also defernating pathway via this hydrolytic O dealkylation. With the unsaturation here, the ether is more reactive, and it can undergo this hydrolytic O dealkylation, and then forming this fluoroalcohol, which underwent spontaneous deformation too, and then the other structure may form this end product TFA, trifluoroacetate. As I mentioned, it's persistent. By quantifying this TFA, we can back calculate how much, about 2 micromolar of the parent compound underwent this pathway. And then another defernating pathway is the hydration pathway. Hydration is occurring at this unsaturated carbon-carbon double bond. And we didn't nail down the detailed deformation pathway, but there were unclear pathways leading the formation of fluoride iron because we detected this transformation product intermediate, this transformation product TP273. Okay, that's the pathway we interpreted for this structure that has both ether and unsaturated carbon. And the difference between the ether plus carbon-carbon double bond to the unsaturated structure only without ether group is this structure on the left only underwent hydrogenation pathway by turning this carbon-carbon double bond to saturated carbon single bond without any deformation activity. But by introducing an ether group here, as highlighted here, forming this structure, and this structure also underwent deformation pathway and with higher biodegradability. So that's the combination of biodegradable moieties in a single structure may further enhance the biodegradability of that PFAS molecule. The implications is now we identified moieties that make a PFAS structure more biodegradable, such as unsaturated carbon, chlorine substitutions, carbon-hydrogen bonds, and an ether group. So those moieties in the PFAS molecule can help us understand different PFAS structures, how they behave, and what their environmental fate could be by understanding which structures may be more biodegradable under which conditions. And it's also helpful for us to track the source of PFAS by detecting signature transmission products. For example, some dicarboxylic acids may be formed from chlorinated substituted PFAS molecules. And if we detected some diacids PFAS, it's likely that it's a transmission product from certain PFAS source structures. And it can also help us to do the toxicity and risk assessment because the PFAS released to the environment originally may be transformed, ending up with a different transmission product. So it is not the original structure that we are concerning about. It is the transformed daughter products that we may want to understand better how they affect the ecosystem and how toxic they are to human health. More importantly is some biological processes may be applied and integrated into the physical chemical approaches to achieve cost-effective PFAS treatment. And how we can handle it is we can use biological treatment process as a pre-treatment or post-treatment to lower the cost. I'm collaborating with Professor Jin-Yong Liu at UC Riverside too, who is a chemist and who is an expert in chemical destruction of PFAS. So we collaborated on this project using his UV sulfide reduction of PFCAs, and then we combine our aerobic biological treatment, just activated sludge, to enhance the total degradation or total deformation performance. Why is the UV sulfide system is not perfect, especially for the ultra short chain PFCA, such as PFPRA, 3-carbon, fully fluorinated carboxylic acid, because there is one byproduct in the UV sulfide system that is very stable. It cannot be further degraded well. So you can see this blue bar represents the effluent of the UV sulfide reactor. Majority is the trifluoropropionate acid, but this structure is the favorable food for the activist sludge because I just showed you this structure can be completely degraded via a beta oxidation-like pathway. So we apply activate sludge treatment after this UV sulfide treatment, and you can see this major stable byproduct in the effluent is gone, is completely degraded. The same for GenX. So GenX is not that easy to be treated either by advanced oxidation or by advanced reduction processes. And the total deformation percentage is so-so, it's nearly half or even less than 50%. By adding a post-aerobic biological treatment, we can enhance and increase the total deformation by about 20%. We have demonstrated it in a batch scale experiment together with other ether fully fluorinated non-biodegradable ether structures. We can use chemical treatment first and then combining with the aerobic biological treatment process to achieve a higher differentiation degree and using a lower cost. With that, I would like to thank you for your attention, and I'm happy to answer your questions.
Thank you, Dr. Man. Very interesting presentation. So, so we still have some time for questions, and you may submit your question via the chat box, or just unmute yourself and speak. We have any question from the audience? Can you go back to slide number 18? Yeah, so here I didn't quite follow, because you show the numbers 26. There's a major 3 different pathway you showed. You are muted, so I can.
Sorry. This is the back calculation. So let's see, we have the standard, this E11, we have the standard compound. So we can quantify this major transformation product. That's 26 micromolar at the end.
Okay.
Yes. We can quantify this TFA, that's how we get this 2 micromolar. And then we quantify the total defluorination number. So if 2 micromolar parent compound underwent this pathway, we calculated, we starting from 7 fluorine in the structure, and we ended up with 3 fluorine. So the total will be 4 fluorine per molecule. And the differentiation will be 8 micromolar. We can quantify the total fluoride formation, which is 26, and then there will be 18. And this pathway, yeah, we interpret this pathway like it's via this differentiation. And then the fluorine that's it's 18 micromolar, and then this 26 is 2, and the 9 is calculated from the degradation of E12, how much E12 is being removed, 37. So the mass balance here is 9. So that corresponding this pathway, there should be 2 fluorine being cleaved, but we don't know the differentiation pathway, the exact differentiation pathway.
Okay.
Yes. So it's a calculation based on the parent compound removal, the formation of the transformation products that we have standard compounds so that we can quantify, and also the quantification of the fluoride ion at the end.
All right. Thank you.
Yeah.
I think we have a question from in the chat box. So the question is, how did you measure PFAS?
Pretty measure PFAS degradation. Well, there's an idea. Well, this is anaerobic system. We haven't tried anaerobic system, but our collaborator at University of Minnesota, they have aerobic system. They have a high throughput fluoride ion detection method, which they published in, I forgot the journal, maybe AEM, and using a colorimetric fluoride ion detection. So that can be used to do a quick screening of PFAS degradation, but that's in their aerobic system, specific medium matrix. So because there may be some interfering factors if you change the medium and if you switch to the anaerobic condition. So we didn't try that. but there is potential. And the representative PFAS chemicals, that's what we use. Yeah. Concentrations, we use relatively high concentration, it's 100 micromolar. For biodegradation, I mean, I forgot to mention the biotransformation pathways. We haven't identified any metabolic pathways. All the pathways or all the transformation activities we found, they are co-metabolic. What I mean co-metabolic is you have to provide a primary substrate such as lactate, methanol, or any other organic carbon to let them grow. So for these co-metabolic pathways, or very low concentrations, I'm not sure how it works, but the idea is high concentration. So treating PFAS is the same thing. We need to concentrate. For example, drinking water sources, if that's contaminated by PFAS, you need to first concentrate it by physical removal, physical concentration, membrane filtration, GAC or resin, ion exchange resin, and to concentrate the PFAS in the sort of concentrate or filtrate, which has a really high concentration of PFAS, so that you can do a centralized treatment. And the same thing for biological processes. Right now, we're using relatively higher concentrations, but after we know better and better of the microorganisms and we discover more and more powerful microorganisms, I don't know. The answer is still being explored. I don't know the limit. You never know the limit, the limitation of microbes, right? You never know the boundary. So that's what we're doing right now, is kind of explore the boundary of microbial deformation, like how far we can go to understand it better.
Do I have any other questions from the audience? Okay, so I think in the very last slides, you mentioned the chemical treatment and combination of chemical treatment and biological treatment. So you mentioned the go with the chemical treatment first and then biological treatment. Is there a way you can like switch, combine this together?
Yes, you can switch. You can switch one versus the other, but using microbe first will be more challenging because usually we start with a fully fluorinated structure. So that fully fluorinated structure may not be effectively degraded by microbes. So if we can find any microorganisms that can help to do the first step, carbon fluorine cleavage, and then we can leave the remaining, the products to, such as advanced oxidation or, other aerobic treatment chemically or even biologically. So it is the very first step. If you're treating, for example, PFOA, PFOS, the first step is the most critical step. The first step is breaking down the first carbon fluorine. And biologically, right now, it's very challenging. I don't think it's doable right now. So that's why the treatment chain system right now we're testing is following with the post-treatment as an idea. But potentially, you could use the microbial at the beginning as we discover effective microbes that can break down the PFCAs or to carry out the first step that we want. And then the other approaches can take over next. So it's theoretically it's doable, but right now the cases I'm showing is just using microbial degradation as a post-treatment because we don't have effective microbes for the first step.
So we have a question from Jenny.
Did you try biodegradation in the presence of other contaminants? Refer to target doses. That's very well, that's possible. I mean, other contaminants, I don't know, depends on what types. If that's like only normal organic carbon contaminants, or I'm not sure what they are talking about because it may be different microorganisms doing things. For example, did you mean chlorinated solvents? Or did you mean hydrocarbon oils? So it's really specific. If that's chlorinated solvents can be degraded by a totally different group of microbes. It won't matter that much, but there are study did show that the presence of chlorinated solvents will inhibit the some processes, some microbial activities. So it'll be complicated. It's what I can tell is the PFAS degrading microorganisms are not the same group of microbes that capable of degrading chlorinated solvents. So they're different. But the chemical, depending on the concentration of chlorinated solvents, may inhibit microbial activities in general because those are toxic. I've done experiments. My PhD did chlorinated solvents biodegradation too. And if you use higher concentration of chlorinated solvents, they may kill certain microbes in the community. So I think I don't have a concrete answer here, but yes and no, depending on contaminants to talk about and the responsible microorganisms that can do the degradation. Yeah, if they're the same or not. So for chlorinated solvents and PFAS, I can tell, to my knowledge right now, what I reported in my talk today, none of those are the same ones as the dihalococoides that has been identified for like TCE degradation or PCE degradation. So they're different. There's one microbes that can be used for the first step is actually, since we're talking about the chlorinated solvents, is the chlorinated PFAS. So microbes can do very well, very good job to chlorinate PFAS. Especially there's more than one chlorine substitutions. Yeah. Thank you.
You mentioned, in addition, chemical treatment, biological, and you mentioned the physical treatment, like using GAC, ion exchange resin, for the enrichment as a first step. So that means for the biological treatment, you need to reach a certain level of concentration first?
It doesn't necessarily to be the case. It's just right now we are using a high concentration. And since that's co-metabolic, we didn't test how low the initial concentration could be for the microbes to start the activity. But when we add this 100 micromolar parent compound, in the end, our instrument cannot detect it. It's not detectable at the end. So it seems like the degradation is pretty complete. So we didn't test, for example, if we start with like 10 nanomolar, 1,000 times lower concentration, initial concentration, what the microbes can do. So it's a common thinking that, or people think if that's co-metabolism, microbes may have a difficulty to handle really low concentration, for example, in drinking water sources. But we are targeting more like a source zone where the contaminant may be at relatively high concentrations in situ, such as chlorinated solvents. And if that's downstream, like in river water, like other surface water lakes, and that I cannot tell that much, but right now what I know is the most effective treatment method is you just do physical removal. Remove it first, and then do destruction. Yeah.
Another question in terms of microbial, the organism you have to grow or culture, can this step be integrated in current like water treatment systems in terms like, okay, I grow something on the resin or others, the physical treatment unit? Would that be something like possible?
It still depends on your goal. As I mentioned, if that's aerobic treatment, the two examples I given in the end, we're just using active sludge, and because it's very easy to get, and the point is like for typical wastewater treatment plants, it can be really easy to be adapted if you want to target certain PFAS treatment. But microbial degradation is there's a feature that's also related to my title, it's a structure specificity. So it really depends on which structures you're talking about. And it's not like one microbial community can be used for thousands of different PFAS structures. So it's really need to be we need to talk in specificity, like very specific PFAS structures that you are aiming to treat. And then you can design certain treatment processes by integrating physical chemical approaches and together with some downstream biological treatment processes to make sure like you're converting the parent compound into like mineralization, complete mineralization, or into some not that concerning products. So yeah, we need to really look into the detailed structure. Integration is yeah, it's always possible. I know there are other researchers investigating the combination of chemical treatment together with biological. It's also active as sludge. It's enriched, like, yeah. So it's possible.
Okay, so my last question is, has your group or any other group looked into the tech economic analysis of this type of technology for the future industrial application compared to other chemical or the physical treatment?
You mean like economic
The cost.
analysis? Cost,
Yeah,
right?
cost
No.
for the industrial application of this technology.
No, I mean there are limitations as I mentioned. It's really structures specified. So there are not many structures that we can handle, and we haven't tested that many structures so far. And I think the point right now is not the cost. The point right now is how to enhance the performance. So we need to reach like 100% or near 100% defluorination first for more than what we tested so far. So we're still exploring more structures and to kind of generalize this approach. I mean, the generality, how general this approach can be applied, even for PFCAs, that's okay. And then second is if we can handle more than PFPRA or more than Gen X. And the second thing is for Gen X, we even didn't reach like 90% defluorination. So we need to enhance the performance first, no matter what the cost is. And the second step is just lower the cost, right? So I think the first thing is I think this should be considered first is the performance. So you need to optimize your technology first to reach like near complete defluorination. And then we talk about the cost. I think that's my order. So.
All right, we have 2 minutes left. If there's any question from the audience, we can take the one last question. I guess if no, let's thank our speaker, Dr. Meng, again. Thank you very much for a nice presentation and nice discussion. Thank you, everyone, for joining us today, and I hope to see you guys in our next lecture. Thank you.
Thank you.
Have a good day. Bye-bye.
Bye.